Okay, so you guys feel free to take out your notebooks. We're going to do our take 5 minutes as soon as it comes up here. Okay, there we go. So you're studying. Oh, by the way, I hope you guys are all studying because we're about a week away from another exam. I know. Just like the syllabus. Not next week, but the week after. So we've got 2 more lectures. Yeah. I know, it's horrible. Okay. Halt. Okay. You are studying for the next exam, and you're studying a new protein. Okay? We're ready? I'm always making you study new proteins. It's called Protein X because I couldn't come up with anything better. You're studying a new protein with unknown function. It has homology to tubulin, so you think it must be involved in microtubule function. You set up an experiment as follows. So you've got 2 test tubes. In tube 1, you put your tubulin dimers plus GTP. In tube 2, you put your tubulin dimers plus GTP plus protein X. You incubate, and then after incubation, you run an experiment where you look at microtubule polymerization. You find microtubules polymerizing at a faster rate in tube 2 compared to tube 1. What is the identity of protein X, or what function does it have? Okay, so take 5 minutes. This is a really great practice problem for the exam. Oh my God. Right. How are people doing? Do we need more time? Okay, sometimes when the chit chat starts, we're only about 20 seconds away anyways. Okay. Who is going to answer today? Jessica. Okay, so just start by telling us how you approach this question, because that's usually the hard part.
Well, there's two different test tubes, and one of them has JTP, the other one has JTP plus protein X. So I'm trying to figure out the identity of that extra protein in that test tube too.
Okay, so you basically broke it down into what the question is,
Yeah.
but weren't quite sure where to go after that. So yeah, you're right. You want to compare the two test tubes and what's in them, but you really want to pay attention to the data, right? And so the data that I've given you in this is homology to tubulin and microtubules polymerize at a faster rate when you add protein X. And so you have to think to yourself, well, what proteins have we learned about that have high homology to tubulin and will affect microtubule polymerization? Yeah.
Gamma tubulin?
Right. Gamma tubulin, right. So it nucleates. So what does nucleation do? Remember that. Let's bring a pen today. I'm just falling apart. Okay. So remember we had those graphs. I'll do it on that side too if you can't see. And there was this kind of growth curve, right? Growth curve of cytoskeletal protein polymerization. And there's this kind of lag phase. And then this nucleation event happens here. And then it takes off, and you've got that exponential growth until critical concentration, right? Right, this would be your lag phase. Oops. And that would be your nucleation. So here's your lag, here's your nucleation, exponential growth, here's your critical concentration. So what this is doing is it is getting rid of your lag phase, right? You don't need to randomly form the nucleus. The nucleating protein is forming the nucleus. It's nucleating the microtubules. So the answer was gamma tubulin ring complex. Did anyone approach this in a different way, got the same answer, and feels like sharing what they thought will be useful to the other students in this class? Okay. Okay, so far what we've done, we've been talking about all the different key players and the major concepts. We went through some toxins and bacterial homologs. That's kind of like stuff for fun. Definitely not the meat. I always get the question when people are studying, which is, what should I know about the toxins and the homologs? Just like understand it, appreciate it. But it's definitely not something you should sit there and commit to memory. And then we started talking about accessory proteins. We started with talking about nucleating proteins. And so as we start to go into the accessory proteins, I want you to stay organized. Like with all studying, if you stay organized and you know where to put all this information, then you'll have no problem learning it. But where if everything feels scattered and you don't know how to fit all the details in, it's very hard to study. And so as we go through the accessory proteins, they're categorized based on function, and usually we have like functional homologs for our actin and tubulin. And so if you were making a chart, it would be a chart where you've got function, nucleating protein, column 1, actin, column 2, tubulin, and you would fill it in kind of as you go, something like that. Or you can have, remember I said you can make your microtubule or your active polymer, draw it out, and so then start drawing in the functions of all the different proteins as we go. So that's just a good way to keep things organized. And it's a good way to be studying now, because you should be studying now for the exam. Okay. So this is where we ended. We're talking about the microtubule organizing center, which is located near the nucleus. So this is the centrosome, and it's covered with more than 50 gamma-tubulin ring complexes. And that's why you have the nucleation of microtubules at this location. Usually you see kind of like a star bursting out from right next to the nucleus. The reason it's called a centrosome is because it ends up being centered by the activity of the microtubules pushing against the plasma membrane. You can imagine if you've got all these hands poking out, pushing against the plasma membrane, it's going to center the centrosome. They duplicate during interphase, I should have corrected that, prophase, and then they move to opposite sides of the nucleus to form the mitotic spindle. So that's where we ended on Tuesday. Right, so that's how it looks. Notice the pluses, that's the plus end, right? So remember we talked about treadmilling and all of that stuff. And this is why I say with microtubules, it's kind of different in the cell because you have your centrosome. And the minus end is busy, so you're not having tons of things leaving from your minus end, right? It's busy with a gamma-tubulin ring complex. Okay, so I just said, I always like immunofluorescence where it applies because then you can visualize things, think about things. So centrosome duplication happens at the beginning of mitosis before the mitotic spindle, right? And so this is just going through the different phases of mitosis, right? So this is interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Ooh, I didn't even have it labeled. So if you follow this, you can see your microtubules are in green, your DNA is in blue, and this yellow here is maskin, which who cares what it is, please don't commit that to memory, but it is a marker for the centrosome. And so you can follow it through mitosis. So here you've got one tiny little dot, you've got all your microtubules radiating out from there. It's right next to the nucleus. And then here, you see there's duplication. There's 2 dots. Then they move apart. 2 dots. You've got a different organization of your microtubules when you're forming that mitotic spindle, right? They're binding, interacting with your chromosomes. Notice your chromosomes are different too, right? Here you've got this uncondensed DNA. Here you've got your condensed chromosomes. And then you have separation of your chromosomes, still, 2 centrosomes. Again, this is the end of the separation, and then as you become 2 different cells, everything kind of goes back. Actually, and really, I guess, maybe the masking isn't there yet. Maybe we can't see it here, but you see we go back to 2 cells and you go back to that interphase organization of your microtubules. Okay? Isn't that cool? I feel like cell cycles are a really fun thing to look at. So just a fun fact, centrosome duplication is regulated by phosphorylation by Aurora A kinase. Right now that's just a fact, which you shouldn't bother remembering. But I would like you to just kind of keep that in your mind, because when we start talking about cell cycle, we'll be talking about Aurora A kinase a little bit. It's involved in cell cycle. So it kind of makes sense that something that's one of the things that's regulated in cell cycle is centrosome duplication, right? Does that make sense? Right? The centrosome shouldn't just duplicate randomly. Random duplication, that's called cancer. Okay, so let's just go right into a practice problem to keep things fun and exciting. Okay, so you are studying cell cycle regulation. You're testing different drugs to identify those that cause cell cycle arrest prior to entry into mitosis, so prior to prophase. And so these are your experimental conditions. You treat your cells in culture with drugs for 12 hours. You've got like a whole bunch of different drugs that you're just testing. And then you assess your drug activity through immunofluorescence. So you look at the cells, and you're using antibodies against the gamma tubulin ring complex, which is the green. So those are those dots, right? And DAPI for the DNA. And so here's one result that you found. So the question is, was this drug active? What does activity mean? Well, you're looking for those that cause cell cycle arrest prior to entry to mitosis. You have to ask yourself, what would cell cycle arrest prior to entry to mitosis look like in terms of gamma tubulin ring complex? Was your drug active? Why are you choosing your answer? What results would you expect if your drug was not active or active? So you have to predict what it should look like. And then what controls are missing? Because there are controls missing for this experiment. Okay, so take a few minutes to talk with your neighbor. I'll circulate, clarify any questions people have. Okay, so you really want to focus on your DAPI, which is your blue, and your green, which is your gamma tubulin ring complexes.
I'm sure. I've never heard of anyone. I can feel that. Oh, wow. They say
Okay. Lots of interesting comments and questions. Okay. I definitely heard a lot of people just chit-chatting about random things. I get it. Sometimes they're really hard. And it's 11 in the morning. So early. Who thinks 11's early? Oh my God. I remember being in college. I remember being so proud of myself for getting to bed by 2. Yeah. But I was, you'd never, if you had seen me in college, you would never believe I got to here. But anywho. So when you are just kind of chit-chatting, ask yourself, is this benefiting me? How have I done on the last 2 exams, and would I like to do that? If you don't know how to approach this, then as I'm walking around, call me over and say, I have no idea how to approach this. challenge and ask me so many things, and then they walk away understanding it better. I will not judge you. I will not think you are dumb. I promise. Because there was no way I could have solved this when I was where you are. Okay? Trust me, because I was still sleeping. I had probably skipped class. No. No, by 400 level, I was on the roll. Freshman year was a disaster. Okay. So, question number 1. Was your drug active? No. Who wants to tell me why they came to that conclusion? Did you see cell division? So there was some confusion as I was walking around as to exactly how the assay is done. And so I kind of clarified it's not a single cell assay, you have to look at the whole population. So it's a dish of cells that you add the drug to and you leave it there for 12 hours. So if there's a block, then they should all be blocked. Okay? You can't say it's partially working because that drug is on all cells, right? So you should see no cell division. What else? What else? How did you integrate the staining for gamma tubulin ring complex into your analysis of the data? Yep. So it can show you what's going on with the mitotic spindle. So if you have how many gamma tubulin ring complexes or how many dots should be in the cell in interphase? How many dots should be in the cell in prophase? 2. Okay. You have a question?
No.
Yeah. Okay. Any other thoughts? Okay, good. So what results do you expect if your drug is active or not active? So if it's active, what should you see? No cell division. Yes. Yeah, because that's when you get duplication of your centrosome, right? Yeah. So go back just for a sec. If you go back and look at this, right? This is interphase. This is prophase. See those dots? First one is interphase. Second one's prophase. Yeah. And then you have 2 for the rest of mitosis. Okay. What would you expect if your drug is not active? Cell division, just different cells in different phases of mitosis or cell cycle, right? Okay, so there was some confusion as I was walking around, and it mostly was due to the fact that controls are missing. So what controls are missing? Actually, all controls are missing here. Yes?
You're not running like an assay without the drug.
Right. So I've got no assay with no drug. So how do I know I even added the drug here? Right? What if, like, I have a freshman in my lab who's never used a pipette before? I'm going to be down on freshmen, I don't know, because they're least likely to have pipette. Never used a pipette, and I have put 1 microliter you need to add, and they didn't do it. And so you're going to get this. So do you say this drug didn't work? That's why you need that control. What other control is missing? Yes?
Control whereas you have a drug that you know stops the division. So you have a positive and a negative.
Right. So you need a positive and a negative control. Your negative control is no drug, and your positive control is a drug that you know works this way to stop cell division prior to protein. And then you would see in that your negative control, you should see pretty much this, and in your positive control, you should see 1 dot in each cell, 1 gamma tubulin ring complex. Okay, good. If you guys have questions when you're doing these, please pull me aside so I can help you learn. Okay. So I was saying how centrosome is called a centrosome because the microtubules are kind of pushing out in every direction, and that's what centers the centrosome. And so centrosome has this kind of center-seeking behavior. And so this is just out of the textbook. a picture of an experiment where one centrosome was put in a plastic well with tubulin subunits, so it's going to nucleate and polymerize tubulin. And so over time, what you notice, so there's your centrosome. Over time, and you can kind of see some stuff radiating out, those are your microtubules. Over time, notice it goes in the center. So that's kind of easy to just think about. If you have equal pushing on either side of all sides of the well, it's going to end up in the center. And so this centering behavior of the centrosome is used for positioning of organelles within the cell. Because remember, these microtubules do something, and one thing that's often happening is organelles are moving around by being attached to motor proteins that are walking along microtubules. Okay, so microtubule nucleation occurs at the center of the cell, right, in the centrosome, where, based on the function of actin in cell shape and movement, where would you expect actin nucleation to occur? Where in the cell? Closer to the edges. Yes. So you should get a lot of actin nucleation at the cell cortex, so close to the plasma membrane. Right, so the actin filaments that are just beneath the plasma membrane, they determine the shape and movement of the cell surface. So this is showing two different cells. And this is nice because there's two types of stains for actin being used in this image. Blue is always the nucleus, so there's your DNA. And so you've got red, which is phalloidin. Remember that toxin we talked about the other day. And so that binds to actin filaments only, so that's polymerized actin. And so you've got all this polymerized actin around the edges. It's like a border, right? And then the green is binding to actin, globular actin, so actin subunits. And so you notice there's this pool of actin just in the cell. And that kind of makes sense with what we've talked about with that dynamic instability and how you've got this growth and shrinkage, has to do with critical concentration, and then all these accessory proteins that are regulating it. Right? So this actin is used for formation of microvilli, filopodia, lamellipodia, phagocytic cups, so all these things that have to do with protrusions from the cell surface and cell membrane. And these things are often regulated by external signals. So we'll eventually tie in signaling. We already did tie in signaling, right? We were talking about Bro and effectin and all that, right? Actin nucleation is catalyzed by one of two different types of nucleating proteins, ARP, which you've learned about in recitation this week, or formins. So as you're making your chart, now you would have nucleating proteins, then you have microtubules. What was that nucleating protein for microtubules? Gamma-tubulin, gamma-tubulin ring complex, and then you would have under actin, ARP complex or formins. Okay, so this is how we start to organize things. The ARP complex is similar in function to the gamma-tubulin ring complex. It nucleates actin filament growth at the minus end, so it binds at the minus end, allowing elongation from the plus end. And this is an important thing to think about because you're going to compare it with formins that work in a little bit of a different way. And so here, here you can see ARP23 is the blue, and what it does is it polymerizes actin. But notice it makes this kind of web of actin, bless you. And so it also, ARP and formins participate in formation of actin that have different conformations and are used in different kinds of functions. So that's another thing to think about. The structures of ARP2 and 3 are similar to actin. That's also similar to what we talked about with the gamma-tubulin ring complex. And it kind of makes sense, because if it's a nucleating protein, it kind of makes sense you should have a similar structure to the proteins that are being nucleated, right? So this is just showing the structures. There's actin, ARP2 and ARP3. Memorize that. Yeah, you're going to have to draw these structures on the exam. Using your mouse on a computer. It's going to be horrible. I can't stand when people make me memorize structures. Anyways, no one makes me memorize anything now. Can't remember anything. Okay. Actin filament nucleation. So here you have ARP complex, and it's not active until WASP. Remember, in the paper for this week, we were talking about ARP and WASP being recruited and being used for pedestal formation, right? Yeah, I see some people in the room. Who had recitation today and has no idea what I'm talking about? Okay. Hopefully between now and then you learn. Did you have a question, or are you really just being honest? I love honesty. I think it's beautiful. Okay. I do. I was telling my daughters today, like, my daughter said, oh, my friend told me that where I live is Ratchy, or something like that. And I didn't know what she meant, so I said, uh-huh. And I was like, you didn't want to say uh-huh to that. And I was like, you need to tell people when you don't know what they're saying, because she could have said, you're ugly. And you said, uh-huh. Which I guess is a way to confuse the person. Maybe that was her tactic. Cute. I was like, no. Just say, I don't know what you're talking about. So, thank you for saying, no. WASP is an activating factor, so it actually turns on our 2 and 3, and then you get nucleated actin. So it binds at the minus end and adds at the plus end. So remember this web that I mentioned before. The ARP complex nucleates most efficiently when bound to the side of another actin filament, specifically at a 70-degree angle. That's weird. And this results in a web of actin filaments. I'm just amazed that someone measured it. That's amazing. So you've got this web of actin filaments. And so nucleation by ARP is associated with formation of these webs, and that's associated with the structures at the leading edge of a cell. And when I say leading edge, that's the edge of the cell that's pushing forward when a cell is moving. It's leading. Questions? Okay, here's another practice problem. We'll just follow them today. You are studying the ARP complex and want to know whether it is involved in formation of the leading edge. You fluorescently labeled antibodies against actin and ARP3. You do immunofluorescence and see the image below. Actin is red, ARP is green. Yellow means they're in the same place. So take a minute to look at it and talk to your friends, and then you'll tell me what you think.
Okay, so. Yeah. So the art complex is in a certain direction. And so yellow is actually so art
Okay, guys. So I just got about 10 people say, I don't really get the leading edge. What do you mean? And so usually when 10 people ask me that, that means around 90 people have that actual question. So the leading edge, if you imagine I'm a cell. What shape did I do? If I'm a cell and I'm moving this way, this is my leading edge, right? The part that's pushing forward, right? Now I have to run this way, leading edge going this way, right? And if ARP and actin, ARP nucleates actin, right, and that's associated with movement, one would expect it would be found in the leading edge. So, what do you see in this picture? You see a leading edge. Where's your leading edge? It's at the top. Why is it at the top? What tells you that? It's yellow. So the other question I got is, what is co-localization? And that means the two proteins are in the same place together. And so then I got a question that was like, but all the ARPs in the middle is green. I'm like, yes, ARPs alone in the middle, but ARPs with actin at the top. So ARP alone. So don't you still need actin to keep the shape of the cell? So that's why you see red everywhere. Red actin is everywhere. But the key here is connecting ARP polymerization, ARP nucleation of actin with formation of these web-like structures associated with movement and the leading edge. Yes?
So is it like a signal, receiving complex that brings ARP to the leading edge?
So is this regulated by signaling? Absolutely. And so as we get to the end, this is all the Rho proteins, actually. These GTP binding proteins that are going to regulate these things. And so, yeah. So as we get to, like, probably Thursday next week, we're going to go through some mechanisms of how movement happens and how it's regulated by signaling and Rho proteins that are involved with formation of different types of actin. Like, so there are these gels or webs of actin that's associated with the leading edge, and those are formed by one kind of Rho, and then there's contractile actin formed by a different kind of Rho. And so then you're going to think back to signaling and what we talked about a few weeks ago, and you'll be like, oh, I get it even more deeply. Yeah. Okay. Other questions? Okay. So gamma-tubulin and ARP proteins are both conserved nucleating proteins. They're conserved. They're similar in structure to tubulin and actin subunits, right? And they may have arisen from gene duplication. And they diverge. So gamma-tubulin is similar to tubulin, ARP is similar to actin, and they may have arisen from gene duplication and then diverged when they acquire those nucleating properties. Okay, so I just said ARP forms branched actin networks. Formins form parallel bundles of unbranched actin filaments. So we have the branching, and then we have parallel, right? Parallel bundles of unbranched actin. And so these are often involved in contractile functions. So my favorite is usually any form of cytokinesis, any form of cell division. You're going to have some kind of tightening around the center and pinching off into two different cells. And so when you've got in yeast, in budding yeast, you have a bud that will grow and then divide. And so here you start to have bud growth. The way to look at this, it's not colored, but the bright spots are formins. Are they formins? No. The bright spots here are actin, I believe. I'll have to look back. But notice it's all forming here, and then it's going to form here, and then it's going to form this contractile actin here, and that's what pinches it into two different yeast cells. And then into dividing mammalian cells, you get this cleavage furrow or contractile ring that forms that participates in cytokinesis. Different functions, right? One is movement, very different from this kind of tightening function. So formins, right? So these different parallel bundles of actin, these are not branched, they're parallel, and they're nucleated by formins. So formin dimers, so they're dimers, and they bind to actin and encourage nucleation by capturing monomers. And they actually bind to the plus end. So the other one bound to the minus end. So formins bind to the plus end of actin and allow elongation from the plus end. So this is different from both ARP and the gamma-tubulin and how they work. There they bind at the minus end. So you can see here what it does is it binds at the plus end and it captures monomers and adds them on. Questions? Okay, so those are our nucleating proteins. So now it's time to have a new category, which is proteins that bind to monomers or subunits. So you've got thymosins. Thymosin binds to actin monomers and prevents their association with actin filaments. So they're not available to be added to growing actin polymers. So the critical concentration for actin is about 1 micromolar in the test tube. Right, so that's the concentration, and that's actin in the test tube. In a cell, the actin concentration is way higher, about 50 to 200 micromolar. Way more actin in the cell. And someone would think, well, if it's that much higher, then you should have constant polymerization of actin, right? But no, because we've got accessory proteins that are regulating things, right? And so what happens is that thymosin binds actin and prevents it from associating with actin filaments. And so you end up with this inactive pool of actin monomers. So what's the advantage to having a pool of actin monomers that can't be active? Who wants to speculate? That's what they always do when you're in graduate school and you're doing your oral exam. They're like, speculate on the and there's no answer. Yes.
Would it change the critical concentration so the polymer wouldn't dissociate on the nuclear end?
Would it change the critical concentration? It does change the effective critical concentration. It has an effect on critical concentration in terms of the dynamic stability of actin. We're going to get one.
If you have it in a pool that you need more monomers, you have access to them.
Okay, so if you have an active pool, then you need them to have access to them. Yep. So if the cell needs to move really quickly, there's just a pool of monomers available. But I just said they're inactive. Huh?
There's going to be some protein that's going to come over.
Yes, there's some protein that's going to come over and change that. Exactly. Right? Because if you have irreversibly inactive actin subunits, that doesn't help anyone. Right. Right. So then there's got to be some other protein that's going to come. Right. And so that's profilin. I love it when my students introduce the next slide for me. Profilin competes with thymosin to bind to actin monomers, encouraging addition to the plus end of actin filaments. So it binds to the end of the actin monomer that would normally associate with the minus end of the actin filament. And so it can readily add to the plus end of actin. And so the cool thing, oh, I think I have it in a few more slides. The cool thing is it actually associates with performance. Yes. Questions, or you just wanted to look at it longer? Yeah.
Is the thymosin bound to the minus end as well?
So your question is it competitive inhibition kind of thing? So years ago, we got the same question in class, which is, do thymosin and profilin have the same binding site on actin? Is that how they compete? And I looked it up, and I got like some inconclusive. We actually had to stump the teacher on that, and it was inconclusive because that's the most obvious mechanism. That means you'll be a great scientist because you immediately went in there and thought to yourself, how does this work? It must be this, right? Very good job. And what I found, I think it was 2 years ago, was it was inconclusive, though that's a really logical hypothesis. You should probably look it up again and see if anything new has come out. Did everyone kind of catch that? If there's competition between thymosin and profilin, how does that work? And is it just they're both binding to the same place, and maybe the numbers of thymosin and profilin molecules in the cell will compete? So if you have more profilin, then maybe it'll bind to displace thymosin or something like that. Yes?
And did you also say that profilin can change the inactive form to the active? Or is it just
Yeah, so essentially, yes. So profilin, what it will do is it'll move the actin monomers from the pool of inactive to a pool of active. Yeah. So probably through some sort of competition. So this kind of shows that. So you can think of it as this kind of cycle here where you've got your actin filament. And so when your thymosin is bound, it inhibits an addition. And when your profilin is available and bound, it'll encourage addition. So there's that competition. And so you can imagine regulating how much profilin is active versus how much thymosin is active is going to determine whether or not you're having rapid polymerization or not. So how does profilin add actin subunits to actin polymers? Well, it actually associates with formins. So if you look at the structure of formins bound to actin, they have these, they call them, in the textbook, they call them whiskers. So basically some protein structure that extends away from the formins that kind of act as a binding site for profilin. And so they'll bind to profilin-bound actin and kind of bring it close to formins and encourage addition of subunits. Any questions? Okay, so if we think about it in a broader context, a more of a big picture context, so we can get out of just thinking about a few different types of proteins, think about it in terms of recruitment of actin to the plasma membrane. Profilin binds to HIP2, which is a plasma membrane lipid, and it helps to localize actin monomers to sites that are going to require rapid actin polymerization. So remember, we talked about how you see a lot of actin at the plasma membrane associated with the cortex, so it needs to be close by. So the fact that profilin is going to bind to these membrane lipids and keep a pool of active monomers available, that seems to be useful, right? And so profilin can be activated in response to some kind of signaling, and then it will compete with thymosin to bind up the actin. And then those profilin-actin complexes will be interacting with membrane proteins. And then you get polymerization of actin all near the plasma membrane, which is where you need that to be happening. Right, so what does this have to do with cell movement? Or what does this have to do with, well, profilin, you're really thinking more about formins. What does this have to do with forming contractile actin or these parallel bundles of actin? Why would you want any of this to happen near the plasma membrane? Speculate.
Always get like movement if you have you push forward. Yeah.
Right, so if you want movement, so I did say that it's mostly ARP, but ARP associated actin polymerization that's involved with the leading edge, but you also have, it's not to say that profilin has nothing to do with that. And then the other thing is when you have your leading edge, you also have to bring up the rear, and that's usually with formation of more of this contractile actin by formins. So again, you're dealing with kind of like scooching up the rear of the cell. That's you need to be moving you need to be interacting with the membrane, right? Any other thoughts? Okay. So this is just a quick practice problem. The critical concentration of actin in a test tube is less than 1 micromolar. However, in the cell, soluble actin monomer is at a much higher concentration. We're talking about this. Which of the following are plausible explanations for this discrepancy? So go through the options on your own or with your friend, and then we'll take a vote. Okay. So, who votes for A? Who votes for B? Who votes for C? And who votes for D? Okay. So there's a little bit of a discrepancy in the classroom between the lovers of B and D. Who wants to tell me why they chose B? Yeah. Okay. Everyone should sit next to Tammy.
No.
Really is. Yeah. Right, so we spoke about how glymosin should be, it binds to monomers and inhibits addition to growing polymers. So that makes sense. Who wants to defend the profilin answer? Wow. Have you been convinced otherwise? Okay, so yeah, so that's right. So this kind of question is a very obvious way to integrate what you're just learning into the exam, right? So it's good to know what each one does. So the answer was B. Okay, Staffman. Staffman serves a similar role with tubulin subunits. Okay, so remember we are right now talking about proteins that bind to subunits and affect their function in some way. We have thymosin, profilin for action, and then for tubulin we have Staffman. So as you make your chart, you can fill it in. All right? So Staffman binds to tubulin subunits and keeps them inactive. Prevents addition to microtubulins. Yes? So this is an accessory. These are all accessory proteins. Everything starting with nucleating proteins, these are all accessory proteins. The reason we call them accessory proteins is they're proteins that regulate the activity of actin, not right the activity, the dynamics of actin and tubulin. That's why we call them, the book calls them accessory proteins. I think that's nice. You don't have to think of them as accessory proteins. You can think of them as just proteins that affect whether or not microtubules are growing or not, or actin polymers are growing or not. Yes? So are you saying since the, tell me if I'm interpreting your question right. Since the Staffman binds to subunits, would you expect it to have the same effect on critical concentration as the other proteins have on actin? The same amount of Staffman? Oh, would you expect to find the same amount of Staffman as tubulin? So like the same concentration of Staffman in the cell as tubulin. And is that because you're thinking you need enough Staffman to bind all the tubulin? What do you guys think? Would you expect to have the same amount of Staffman and tubulin?
A lot of tubulin is going to be used from the assumption that keeping the cell shape. So, I mean, we do need excess, but you wouldn't need as much.
Okay, so hypothesis number one, you wouldn't need as much Staffman because a lot of the tubulin's busy doing microtubule. Yeah.
If there was the same concentration, the microtubules wouldn't be growth.
So if there was too much Staffman, maybe it would inhibit microtubule formation, period. Okay, that's a good hypothesis. I saw another hand up in the back. Yep. How about over here? I would guess, well, there's two things going on. One is there doesn't seem to be a need for equal numbers because so much of your tubulin subunits are in microtubules. There should be some sort of concentration available, but don't forget every single gene regulated and expression is going to fluctuate based on the needs of the cell, so you can always make more. And the other thing is that Staffman is negatively regulated by phosphorylation. So that does allow for there to be an excess of Staffman that's not needed because its presence isn't just going to bind to tubulin. So there's kind of two things there, right? Activity based on phosphorylation and expression level that are going to determine things. Any questions or thoughts on that one? Okay. So how does Staffman affect the critical concentration of tubulin? It decreases the critical concentration. It definitely decreases the availability of tubulin. So you can have a much higher concentration of tubulin in the cell, but it's not available. How does phosphorylation of Staffman affect microtubule dynamics? I hear it would promote microtubule formation. Yeah, because you're taking some Staffman out of the pool of inactive Staffman. You're taking tubulin out of the pool of inactive tubulin when you phosphorylate Staffman. Okay, so we'll end on this exciting thought. And we'll start with this on Tuesday. Let me just stop this.